Hi, and welcome to segment 2 of the lecture material on metabolism. We are going to pick up with slide number 66 and go through the end. In the first segment, the previous segment on metabolism, we covered the first 3 bullet points here. And in this lecture segment, we'll pick up with lipid metabolism and we'll go through protein metabolism, and that will be it for metabolism. So lipid metabolism. Lipid metabolism includes all of the ways in which your body uses lipids, your body stores lipids, your body makes lipids, and your body controls where, when, how lipids are used. And so we need to visit these two terms, exogenous and endogenous. So exogenous means from outside the body, and endogenous means from within the body. So exogenous lipids are lipids that you eat. They come into your body, and as they are absorbed across the wall of the GI tract, they end up in chylomicrons, which hopefully is a term that you remember from the digestive system. So exogenous lipids are dietary lipids, lipids that you have recently ingested, and they are in your lymph and in your blood in the form of chylomicrons. Endogenous lipids are lipids that have been manufactured inside your body, lipids that are being distributed. They are on the move from one part of your body to another. And there are lipids that are on the move as well, but they are intended to be eliminated from your body. So we are going to talk about both exogenous lipid pathways. Well, what does your body do with the lipids that you just ingested? And endogenous lipid pathways. Where are lipids manufactured? Where are they stored? Why are they sent from point A to point B around your body? And to talk about lipid metabolism, we need to take a pretty detailed look at lipoproteins. It's a good name. A lipoprotein is made out of lipids and proteins. And what a lipoprotein is a lipid transport pod. You remember that oil and water do not mix. You remember, I hope, that like dissolves like, and lipids are hydrophobic, and your body fluids are mostly water, and water is obviously hydrophilic. And so in order to deal with lipids in an otherwise mostly watery cellular environment or body, we need to handle the lipids in a special way, or else whenever we have free lipids floating around, they'll find each other, and these tiny little lipid molecules will find each other and form big lipid blobs, just like the lipids find each other in your salad dressing after you put it back down on the table. So lipids travel in our watery solutions in this form, a lipoprotein. And what a lipoprotein, it has a very basic structure. At the core of a lipoprotein, the interior of a lipoprotein, we find lipids. And so this might be cholesterol, it could be fatty acids that have come in with the food that you're eating. It could be phospholipids. Any type of lipid or fat-soluble vitamin is going to occur in the interior. And then look at the shell. The shell is a single layer of phospholipids. And remember, those phospholipids are amphipathic. So their little hydrophobic tails are facing the interior, that core, and their hydrophilic heads are facing the watery medium that's surrounding them. Either it's interstitial fluid, or it's cytosol, or it's blood plasma. And so this shell is a monolayer of phospholipids with proteins embedded in it. And these proteins are called apoproteins. Apoproteins occur in the shell of lipoproteins. And apoproteins are docking proteins. They're like little molecular addresses, right? Because these lipoproteins are lipid transport pods, and so they are en route to a particular destination, and the apoproteins determine that destination. These apoproteins will interact with proteins on a particular cell type in the body, and that will enable this lipoprotein to dock in that tissue and unload its lipid-rich content. There are many different types of lipoproteins in the body, and we categorize them by their density. So let's start looking at these different lipoproteins, and here they're lined up based on size as well as density. So lipoproteins are a combination of lipids and proteins, and lipids are far less dense than proteins. Just like oil floats on top of water, it's because lipids are less dense than water. So we have to think about the lipid to protein ratio. And chylomicrons, the ones all the way on the left, chylomicrons are those lipoproteins that form in your intestinal epithelial cells following the absorption of lipids and fat-soluble vitamins and fat-soluble phytochemicals. And they are obviously the largest of all lipoproteins. And they also have the lowest density. And this is because if you compare the amount of lipid to the amount of protein that makes up the lipoprotein called a chylomicron, there's a lot more lipid compared to the amount of protein. And so they are particularly low density. And if we move from left to right through the other types of lipoproteins, we only see their initials there. But let me just read through them so you get a sense of density. So we have chylomicrons on the left. Next to the chylomicrons, we have very low density lipoproteins. That's what VLDL means. And as we go smaller in size, we change the ratio of lipids to proteins, and proteins are more dense than lipids. So as our lipoproteins get smaller, the proportion of protein gets higher and the density gets lower. So from VLDL, we hit IDL, and those are intermediate density lipoproteins. And then we hit the LDL, that's a low density lipoprotein. And then we hit the HDL, that's a high density lipoprotein. And you might recognize those terms, LDL and HDL, because they're part of a cholesterol panel, which we'll talk about in just a moment. Okay, back to exogenous lipids. Remember, exogenous lipids are lipids that you have recently ingested. They've been absorbed, and they are in the form of lipoproteins called chylomicrons. Those chylomicrons started out in your lymphatic system, and then they got dumped into venous blood at the subclavian vein. And so somebody who's recently eaten, they are in the absorptive state, they have chylomicrons in their floating around in their blood plasma. And so what happens to these chylomicrons, these fats that you've just absorbed? These chylomicrons have apoproteins. All lipoproteins have apoproteins, these docking proteins, as part of their shell. So those apoproteins determine the destination. And so chylomicrons, these dietary lipids, are destined, they are addressed, so to speak, to be dropped off at your liver cells or your fat cells, your adipocytes. And so as those apoproteins interact with surface proteins of liver cells or fat cells, the lipid-rich contents of those chylomicrons slip out of the shell and across the plasma membrane of the hepatocyte or the adipocyte. And so those recently absorbed dietary lipids enter into a liver cell or a fat cell. And that chylomicron is no longer a chylomicron. Just the surface phospholipids and proteins are left, and its density has increased. So let's take a look at this diagram. Starting on the left, if you need to, go ahead and review how chylomicrons are formed in your intestinal epithelium and how they get from your intestinal epithelium into your blood. because that's where we start with number 1, arrow number 1 on the left. We have a chylomicron. It's traveling as part of blood plasma, and as that blood passes through a capillary bed in adipose tissue, those apoproteins can allow for those dietary lipids to be unloaded and loaded into fat cells for storage. Then the shell of that chylomicron, the chylomicron remnant, will travel and dock with liver cells, and the liver cells will take in the shell of that chylomicron and the phospholipids and the apoproteins and make use of those molecular building blocks. Those molecular building blocks will be recycled. So endogenous lipid pathways involve lipids that were already in the body. So now we're no longer talking about chylomicrons, but we're talking about the other lipoproteins, the VLDLs, all the way to the HDLs. And so these are lipids that have been manufactured, or they were otherwise already present in the body, and they need to be distributed. They need to be sent from point A to point B, either because your cells need some lipids, or your body has enough lipids, and these lipids need to be excreted as a waste product. And so it's possible for your liver to have excess lipid that needs to be sent to your fat in order to be stored. It's also possible for your liver to generate, to synthesize, to manufacture new molecules of cholesterol, and cholesterol is an important component of cell membranes. It helps keep our cell membranes bendy and pliable and fluid. And so if your cells need cholesterol, your liver will manufacture that cholesterol and ship it out in the form of a lipoprotein. And so this is a diagram showing some of the activities that are part of the endogenous lipid pathways. And so we'll start again on the left side of this diagram, where we have the liver synthesizing cholesterol and packaging that cholesterol in the form of a VLDL. That's a very low density lipoprotein, and of course it has docking proteins. It has apoproteins, I should say. And so maybe that cholesterol is being sent to adipose tissue. Maybe that cholesterol is being sent to some other body tissue. Maybe skeletal muscle cells need more cholesterol to be part of their plasma membrane. The apoproteins help that lipoprotein dock and unload its content. So it's like a little shipping container. And then once the lipids have slipped out of the lipoprotein now has changed density. So we've moved from, on the left, a VLDL, a very low density lipoprotein. Now that we've lost some lipids to adipose tissue, we have a higher ratio of protein relative to lipid, and we have an IDL. So that's arrow number 4. We have an IDL, an intermediate density lipoprotein, and that will continue on its pathway in the blood, and a different apoprotein will allow it to dock to a hepatocyte, a liver cell, and any remaining lipids might be unloaded into that hepatocyte for storage or other processing. And notice that that ILDL, now we're looking at from arrow 6 to 7 to 8, that IDL changes density again to become an LDL, a low density lipoprotein. And maybe that low density lipoprotein is delivering a package to skeletal muscle cells, and that LDL also has apoproteins, and the right receptors are on the exterior surface of that skeletal muscle cell, and that skeletal muscle cell will get a delivery of lipids. So perhaps you've had a cholesterol panel performed. It is part of a routine blood workup, and a cholesterol panel will measure the level of cholesterol in the blood. And of course, cholesterol is part of lipoproteins, and there are many different types of lipoproteins. And so a cholesterol panel includes a measurement of total cholesterol, which is free fatty acids. Those are the triglycerides shown there, plus the various types of lipoproteins, the high density lipoproteins, the low density lipoproteins, and then some tests even separate out the intermediate and the very low density lipoproteins. And you might be familiar with LDLs as, quote, the bad cholesterol, and HDLs as, quote, the good cholesterol. We are still trying to understand the relationship between circulating levels of cholesterol, how much cholesterol is in our blood in its various forms, and your risk of cardiovascular disease. And maybe 20 years ago, we, the body of science, used to think that, oh, if you have a bad cholesterol profile, if you have too much LDL and not enough HDL, then you have an elevated risk for cardiovascular disease, specifically atherosclerosis. And while that still is believed to be true, we the body of science, currently recognize that the role that cholesterol and blood cholesterol play in the body, it's complex and it's not cut and dry, and we're not really sure about the relationship between these various lipoproteins, these various formats for cholesterol, and the risk of cardiovascular disease. We are certain that statins seem to be a really good thing for a whole lot of people. So statins, like Lipitor, statins are one of the most commonly prescribed classes of drug worldwide. And statins, there are many different ones on the market, and what they all have in common is they, by one mechanism or another, they suppress the liver's ability to synthesize cholesterol. Now, you need cholesterol. You need it it's so vital to life that your liver can make it, right? You don't need to eat cholesterol. Your liver can make it. So you can be a vegan, and that's fine. Your liver can make all the cholesterol you'll ever need. But some people seem to have a genetic predisposition to making too much cholesterol, and that does seem to elevate their risk of cardiovascular disease. So several decades ago, statins came on the market, and they seem to be really effective at reducing the risk of cardiovascular disease in lots of people, which is why they are so commonly prescribed. There has emerged over the decades of people taking statins, we've learned something else. They also seem to lower the risk of cancer. Surprise! And what a great surprise. And again, the jury isn't in here. There's not really a scientific consensus. And of course, the story is complex and nuanced. But how cool is that we prescribe this drug to treat one of the leading causes of death in the US. Actually, cardiovascular disease is the leading cause of death in the US, followed by cancer. And we start prescribing one class of drugs to prevent cardiovascular disease, and we end up suppressing the development of cancer. It's pretty cool. And as always, the story is not finished in science. As more and more evidence comes in, we'll get a clearer picture of the relationship between statins and cardiovascular disease, and statins and cancer. Here are some new vocabulary words for us related to lipid metabolism. Lipogenesis. The word says what it means. It's the genesis or the synthesis of new lipids. So lipids play all sorts of roles in your body. Lipids form a class of steroid hormones. Lipids make up cell membranes. Cholesterol is a lipid, etc. And there are two types of cells in your body that are able to manufacture new lipids, and these would be endogenous lipids from within the body. So your liver cells and your fat cells can start with a non-lipid molecule like glucose or certain amino acids, and they can convert that non-lipid molecule into a lipid. And that process is called lipogenesis. And one of the reasons why our liver cells and our fat cells engage in lipogenesis is so that we can store excess energy in the form of fat. Fat has low density relative to the other types of molecules in our body. And fat, maybe you've taken nutrition, and maybe you remember that if we compare a gram of carbohydrate, a gram of protein, and a gram of fat, which one has the greatest amount of chemical energy? Which one has the most calories? The gram of fat. A gram of fat has more than twice as many calories compared to a gram of carbohydrate or a gram of protein. Carbs and protein each have about 4 calories per gram, and lipid or fat has 9 calories per gram. And so we're animals, we move around. Every time we move, we have to carry all of our body weight with us, and that costs energy. And so it's way more efficient for animals, these mobile organisms, unlike plants, these mobile organisms, to store excess energy in the most calorie-dense, lightweight form possible. And so we can store excess energy as fat by converting excess proteins, meaning excess amino acids, and excess carbohydrate, meaning excess glucose. We can convert those molecules into triglycerides and store them as fat. The opposite of lipogenesis is lipolysis. Again, another great word. The word says what it means. Lysis means splitting. Splitting lipids is lipolysis. So sometimes we need to take a triglyceride and chop it up into pieces because we're going to use those pieces. And so shown below, we see a fat cell. We see an adipocyte. And the major purpose of a fat cell is to store fat. And so that yellow blob of triglyceride, that's like an oil droplet inside of that cell. And when those triglycerides are catabolized, they are catabolized into their building blocks, which are fatty acids and glycerol. And that process is known as lipolysis. And lipolysis occurs because we need to liberate those molecules for some reason. We need glycerol, we need fatty acids. It turns out that glycerol can be used for gluconeogenesis. If you're carbohydrate deficient, don't worry about it, you have fat. And we can perform lipolysis, generate glycerol, and glycerol can be used to create new molecules of sugar. So you stay alive, hooray! Another reason for lipolysis is we need to tap into your fat stores as a source of energy, not just a source of glucose, but also to use those fatty acids to directly make ATP. So glycerol can be used for gluconeogenesis, which can be used to make ATP. Those fatty acids can also be used to generate ATP. So this is pretty much what I just said on the previous slide. The products of lipolysis can be used for cellular respiration. There are 2 products of lipolysis. There are fatty acids and glycerol. The glycerol can be converted into glucose. And in the previous lecture recording, in segment 1, we talked extensively about how glucose is used as a fuel source for making new molecules of ATP. The fatty acids can also be used for cellular respiration. And the first step is to undergo a process known as beta oxidation. And then those fatty acids will be converted into a form that can enter into the Krebs cycle, which occurs inside mitochondria. So let's take a look at this diagram and start with the things that we have talked about previously. So what have we talked about previously? Previously, we talked about all of this, actually. So we covered this in the last lecture We talked about using glucose as a fuel source for cellular respiration, and we see the first 3 steps of aerobic cellular respiration. Glycolysis is sort of in that peach box. Glycolysis gives rise to pyruvic acid. And the second step is the formation of acetyl coenzyme A. And the third step is the Krebs cycle. Not shown here, but following the Krebs cycle, our fourth step is the electron transport chain. Okay, so let's add on to that. We are talking about lipolysis. We're talking about how do we use endogenous lipids for cellular respiration. So let's say these are some triglycerides, some lipids inside your fat cells, and you know what? They get mobilized. You're spending more energy than you're taking in, and we need to rely on your fat stores in order to provide the fuel for generating ATP. So it's not really labeled here, but if I could create a label for these arrows right here, I guess the green arrows going from triglycerides to glycerol and from triglycerides to fatty acid, those green arrows represent lipolysis, the splitting of lipids, and then glycerol and fatty acids are the 2 products. Both glycerol and fatty acids can support cellular respiration. They're both fuel sources, but in different ways. So glycerol can be converted into glucose, and then it enters into cellular respiration that way. Fatty acids are not converted into glucose, but rather these fatty acids undergo a process known as beta oxidation, and that gives rise to acetyl coenzyme A. So the energy that was, let me change colors here, the energy that was originally in these fat molecules that were stored in your adipocytes, that energy gets converted into ATP 2 ways, glycerol and fatty acids. A byproduct of beta oxidation of fatty acids are ketones. And that's a whole class of organic molecules, if you remember that from chemistry. Ketones form from the beta oxidation of fatty acids, and it's a normal byproduct of metabolism. We're using fatty acids every day to make some ATP. Yeah, our cells like glucose. That glucose is the preferred fuel source, but there is some flexibility there. It's not all glucose all the time. There are some amino acids get used, some fatty acids get used, and so every day we're producing some ketones as part of our normal cellular metabolism. Under some circumstances, we are producing more ketones than typical. When we are burning more fat as a fuel source for creating ATP, we generate ketones at a faster rate. And those ketones can possibly accumulate in our blood. When we have an elevated amount of ketones in our blood, that's called ketosis. And it turns out that ketones can influence the pH of our blood. They can lower the pH of our blood. So if we are undergoing large-scale beta oxidation of fatty acids, which would occur when we are in a super fasting state. So it's not like you just wake up in the morning and you haven't eaten since yesterday, like you didn't eat breakfast yet. It means you haven't eaten for a day or 2 or 3, or maybe it's a starvation condition and you're really burning your body fat in order to stay alive as a fuel source for ATP synthesis. Your cells are going to be generating more ketones, and those ketones can accumulate. You can create ketones faster than you can get rid of them under those conditions, and that puts a body at risk of ketoacidosis. So remember way back in the respiratory system, we talked about acidosis and alkalosis. When the blood pH becomes too high or too low. Yeah. Ketoacidosis is a form of metabolic acidosis. So your respiratory system is functioning just fine. If anything, your respiratory system is helping to fix the problem. But the problem, the source of the extra acid, is your metabolism, ketoacidosis. So here are 3 common conditions leading to ketosis, having an elevated concentration of ketones in the blood. As I mentioned earlier, starvation, right? So there are no new incoming nutrients, and your body is in the post-absorptive state and trying to maintain homeostasis using the molecules that it has on hand. The same thing is true for a carbohydrate poor diet, like the South Beach diet, the Atkins diet, or the ketogenic diet. They're all variations on a common theme. You dramatically reduce the amount of carbohydrates that you intake. Either starvation or a carb poor diet leads to a lack of glucose as a fuel source. And so the body turns to fatty acids as a fuel source for ATP synthesis, and that results in the formation of ketones. The excessive formation of ketones will elevate the concentration of ketones in the blood. These ketones will spill out into your urine, and sometimes you can smell it in a person's urine. A person's urine might smell sweet. Their breath might smell sweet. It's very easy to test urine for ketones, and you can buy ketone dipsticks in a pharmacy or, of course, online. And somebody who's trying to lose weight using a super low carb diet, they are seeking a state of ketosis. It means that their diet is effective. And then the third condition that can lead to elevated blood ketones is diabetes. And in the previous lecture, we talked about blood glucose homeostasis, and we didn't really talk about diabetes, but diabetes is a disorder that involves the body's inability to effectively regulate blood glucose. So there are different types of diabetes. There are 3 different types of diabetes: type 1, type 2, and gestational. And diabetes is the most common endocrine disorder by far. At this point, I ask in a face-to-face class, I ask people, raise your hand if you know or love somebody who has diabetes. And the vast majority of hands go up in the room. In a classroom full of 65 people, maybe like 3 people have their hands not up. And I tell everyone, if you don't know anyone with diabetes yet, you will. It's that common of an endocrine disorder. And the most commonly diagnosed form of diabetes is type 2. And so it doesn't matter the form of diabetes. What all diabetes patients have in common is that their cells are unable to, for one reason or another, their cells are unable to respond to insulin. And in the previous lecture, we talked about how insulin makes body cells more permeable to glucose. It makes it easier for the glucose that's in the blood to enter into our body cells, and then our body cells are going to use that for ATP synthesis. And in the absence of insulin, or if our cells aren't responding to the insulin that's present, our cells don't get the glucose. And so our cells increasingly rely on alternative fuel sources like amino acids, like fatty acids, for supporting ATP synthesis. And so sometimes we can have undiagnosed diabetes. So somebody is a diabetic, they don't know it, or they know it and they don't control it. They can go into what's known as ketoacidosis. Their blood pH becomes too low, and it's acidosis. It could lead to death if untreated. Okay, protein metabolism. So I hope you remember that proteins are large globular molecules that are made up of amino acid building blocks. They have very specific 3-dimensional shapes, and some proteins change shape as part of their normal functioning. We've been talking about proteins the entire semester, and unlike the other biological molecules, carbohydrates and lipids, proteins aren't stored. There is no storage form of protein in our body. So the amino acids that come in with each meal, they need to be put to use one way or another. There's nothing like a fat cell for proteins. We don't have that type of storage unit. So how do we put these amino acids to use? What do we do with them? One thing is build new proteins, and that's called protein anabolism. And protein anabolism is protein synthesis, right? Anabolism means starting with smaller molecular building blocks and using those building blocks to build larger molecules. So when we use amino acids to build proteins, that's protein anabolism. And we talked about protein synthesis very early on in the semester. And I hope you remember that a gene is a segment of DNA that codes for a strand of RNA, messenger RNA, and messenger RNA is a strand, it's a nucleic acid that codes for the synthesis of a particular protein. The ribosomes slide along the messenger RNA and tack together amino acids in a very specific sequence, and then that polypeptide chain folds to create this new protein. And so our cells are very busy. You might be feeling very lazy, but on a cellular level, you're quite busy. And all your cells, except for your red blood cells, because they don't have any nuclei, all your cells are constantly building new proteins to replace the existing proteins. There's this cycle, there's this protein turnover that's happening inside all of our cells. And so some of the amino acids that came into your blood from the last time you ate, some of those amino acids are being used right now to build new proteins in your body, new enzymes, more hemoglobin, etc. I hope you remember that there is a catalog, a library, an alphabet, so to speak, of 20 different amino acids that are used for protein synthesis. We also use amino acids in other ways. Certain amino acids can be used for gluconeogenesis. They can be used to create new molecules of glucose. Some amino acids can be used for lipogenesis. You can take amino acids and convert them into lipids. Your liver cells can do that. Your fat cells can do that. And then some amino acids can feed right into that Krebs cycle and be used to support cellular respiration. It's a very simple diagram. It's a great diagram. So let's start with from protein to amino acids. When we go from protein to amino acid, is that anabolism or catabolism? That's catabolism. Let me get the pen out here and show you. This is protein catabolism. Catabolism, like cut. When proteins are cut apart into amino acids. And then this is using amino acids to make proteins. This is protein anabolism or protein synthesis. And so these amino acids are really versatile, right? They can be used to make proteins, but they can also be used to make glucose. They can be used to make lipids. They can be used to fuel cellular respiration, to make ATP. And so when we use amino acids, when we use these little building blocks, the byproducts of amino acid use, and it depends on how we're using them, but the waste products are urea and carbon dioxide. So carbon dioxide we talked about previously. Carbon dioxide is produced during cellular respiration. Just like when we use glucose as the fuel source for making ATP, carbon dioxide is a waste product, right? That carbon from the glucose has to go somewhere. But you know what? Amino acids have carbon as well. And so when we use amino acids to fuel ATP synthesis, a byproduct is carbon dioxide. When we use amino acids for generating new molecules of glucose, for ATP synthesis, for generating lipids, we create this waste product called urea. And urea has come up for us a couple of times. Urea came up in the context of the urinary system, right? Urea, urinary system. That's not a coincidence. Urea is a component of urine. And it's how our body gets rid of this excess nitrogen that is the product of really amino acid metabolism, which is technically protein metabolism. So let's take a look at what's going on here in the liver. Let's start up here with an amino acid, and I hope you recognize that this is a generic amino acid. It's got all the foundational building blocks, right? There's that central carbon atom here, and to that central carbon atom, we have a carboxyl group is attached on one side, an amino group is attached on the other. There's a single hydrogen atom attached, and the fourth chemical bond, the fourth thing attached to that carbon is the variable side chain. So this green, I don't know, it looks like a green hot dog. This green hot dog is what is going to define one amino acid versus another. Is this a serine or an isoleucine or an arginine? It doesn't matter for our purposes here. So there's an amino acid in your blood, and it gets taken up by a hepatocyte, by a liver cell. This brown blob right here is the liver. It gets taken up by a liver cell, and that liver cell is going to use that amino acid for something. It's going to use that amino acid to make ATP, or maybe make new molecules of lipid, or maybe make new molecules of glucose. No matter what that liver cell is going to use the amino acid for, that amino acid needs to be deaminated. Deaminated, which means to take the amino acid off of, let me back up. Deamination means to remove the amino group from an amino acid. So at point number 1 right here, we're starting with our regular amino acid and we're stripping this amino group off right here. And this amino group can very easily combine with another hydrogen ion to form, you know what this is? Ammonia. And ammonia is toxic. It's a strong base. So if ammonia were to be allowed to accumulate in your blood, it would very quickly disrupt your blood pH. It could potentially disrupt the pH gradient inside of our mitochondria. Do you remember that? That very high hydrogen ion concentration gradient inside the mitochondria that gives rise to or allows ATP synthase to make so much ATP. Ammonia could potentially disrupt that gradient. And so there's always a little bit of ammonia in your blood and in your urine, but most of the ammonia that forms from deamination, when we want to use these amino acids, most of that ammonia gets converted into urea. And this largely happens in your liver. And urea is a non-toxic, water-soluble, neutral pH substance. And so in your blood, you have both ammonia and urea. And most of the nitrogen that your body needs to excrete is in the form of urea. Some amino acids can be used to fuel cellular respiration. So this big green circle represents the Krebs cycle, also known as the citric acid cycle. And so previously we had talked about using carbohydrate, specifically glucose, to create pyruvic acid and so on, right? Glycolysis, the product of glycolysis is pyruvic acid. But this diagram focuses on all the different amino acids that can feed into, meaning they can be the starting ingredient for the Krebs cycle. And so certain amino acids, so all of the words in like this light yellow tan box, all of these light yellow tan boxes, all of those words are individual amino acids. And so some amino acids can be converted into pyruvate and then get converted into acetyl-CoA, enter the Krebs cycle. And other amino acids can enter the Krebs cycle at different points. And the purpose of entering the Krebs cycle is to serve as an energy source to support really the electron transport chain. Not shown here, but let me remind you that one of the important products of the Krebs cycle is that NAD and FAD get electrons, and they will transport those electrons over to the electron transport chain, which is also inside the mitochondria. PKU, phenylketonuria. Maybe you've read a food label at some point in your life, and you remember reading, it's often on yogurt containers and what else? Sugar-free gum. It says attention phenylketonureics. This product contains phenylalanine. So phenylalanine is one of the 20 amino acids that our cells use to synthesize proteins. And PKU is a genetic metabolic disorder. So a person is born with it, and they lack a particular enzyme that allows cells to use the amino acid phenylalanine as a fuel source for cellular respiration. So people with PKU, they don't produce a particular enzyme. That enzyme is called phenylalanine hydroxylase, and it disrupts this part of the previous diagram. So phenylalanine in most of us can be converted into tyrosine, and tyrosine can then be used to fuel ATP synthesis. But in people with PKU, this step doesn't happen with the red arrow. That step doesn't happen. And so what ends up happening is in the body of somebody with PKU, phenylalanine can accumulate. It builds up. And it turns out that this leads to problems, big problems in the central nervous system. So if it goes undiagnosed, a newborn with undiagnosed PKU can develop severe developmental disabilities, severe cognitive impairment. The child will never lead anything close to what's considered a normal, typical, healthy life. And so PKU is routinely screened for in countries all over the world, because if we can identify it at birth, well, it's pretty easy to deal with. Although I say that as somebody who doesn't have PKU, apologies. The way to deal with PKU is to limit your dietary intake of phenylalanine. And there are databases and databases of food sources of phenylalanine. And so the person just manages their diet. Phenylalanine is never allowed to accumulate, and they never suffer any of the developmental disabilities that come from growing up with too much phenylalanine. Here are some closing thoughts about metabolism. In the previous lecture, more so than in this lecture, we've been thinking about energy transformation, how the chemical energy in the food molecules that we eat get converted into the chemical energy in molecules of ATP. And ATP fuels all biochemical processes. And that's true for all forms of life on the planet use ATP. Animals, all animals, are solar powered. The energy that's passing through your body right now, the energy that animates you right now, literally came from our sun. And look at the equation, the summary equation, the bidirectional summary equation at the top of this page. I hope it looks familiar to you. In going from right to left, that's the summary equation for cellular respiration, where we use glucose as the fuel molecule. So on the right side of that equation, we see glucose combines with oxygen. You know what that oxygen is needed for. That glucose combines with oxygen. That glucose ultimately gets broken down. Those atoms get busted apart to form molecules of carbon dioxide, water, you know where that forms, and that energy from the original molecule of glucose gets captured into the formation of new molecules of ATP. So that's cellular respiration. Notice how perfectly it is the opposite of the summary equation for photosynthesis. Plants, green things, do photosynthesis. where they use the carbon dioxide from the atmosphere and water and the energy in sunlight to make new molecules of glucose, and they give off oxygen. And so you are part of this system. And so just, thank a plant or algae. Thank anything green, because it's really fueling our whole biosystem. So this brings us to the end of metabolism. In segment 1, we talked about the first 3 bullet points here. And today, in segment 2, we talked about lipid metabolism and protein metabolism. See you next time.